Hello everyone, I'm Kavita from first year MSc Botany. Here I'm going to present my e-content regarding microbial differentiation. Microbiologists can identify the microbe by its morphological features and document all of the organisms in the sample. Differentiating bacterial strains within a single sample can be a daunting challenge. Microscopic studies of the example can be used for simple identification and differentiation tasks. Staining, culturing, and simple biochemical tests are other traditional methods for distinguishing between the microbial species. There are two methods for differentiating the microbial species. One is traditional method and another one is modern method. Microscopic features can be used to identify bacteria include all over the shape of the microbe. Microbes can be shaped like rods, spheres, or spirals or other shapes. Gram staining is very common test used to determine the type of bacteria in a culture. It was named after Danish bacteriologist Hans Christian Gram. Species including Bacillus, Staphylococcus, Streptococcus, and Clostridium turn purple with gram stain. Other species like Escherichia, Helicobacter, and Salmonella are gram negative, meaning they don't change color with gram stain. Some examples of biochemical testing include catalyst testing, oxidase testing, and substrate utilization test. In the modern method, more sophisticated method for differentiating the microbes, it include PCR-based identification, microarray-based identification, and immunological identification. In PCR-based identification, real-time PCR is used to amplify certain genes, which are then sequenced to confirm the identity of the microbes. In the microarray-based identification, microbial DNA sequences are hybridized to species-specific oligonucleotide probes in a grid format. The identity of microbe can then be determined by finding the grid of the positive result. An immunological assay can detect species of microbe by linking antibodies for species-specific protein to a detection method such as dye. Methods like fatty acid profiling and chemoprofiling can also distinguish different microbes from each other. Microbial species often have unique combinations of fatty acids or metabolites that can distinguish them from each other. The exact definition for microbial differentiation is, it is the modification of a cell in terms of structure or function occurring during the course of development. There are different types of differentiation in the microbes. It includes in bacteria, virus, fungi, and protozoa. First, we are going to see about bacterial differentiation. In the bacterial differentiation, the cytoskeleton, it is a cellular structure like a skeleton contained within a cytoplasm. The differentiation of bacteria based on cell wall composition. The cell wall is the outermost layer of the cell, and it gives protection to the cell and it maintains the shape as well as the osmotic integrity. It provides structure and shape and protects the cell from osmotic forces. Consisting of three morphological distinctions that is found in the bacteria. Bacteria can be classified into two groups based on their cell wall, gram-positive and negative. Most have cell wall composed of peptidoglycan, and gram-positive cell wall consists of thick layer of peptidoglycan and unique polyalcohol called teichoic acid, some covalently linked to lipid, forming lipoteichoic acid that anchor peptidoglycan to cell membrane, retain crystal violet dye in gram staining procedure so appear purple. Gram-positive cell wall is referred to as a peptidoglycan layer, and it is composed of alternating subunits of two carbohydrates, N-acetylmuramic acid and N-acetylglucosamine. The gram-positive cell wall is referred to as a peptidoglycan layer, and it is composed of alternating subunits of two carbohydrates. Gram-negative cell wall. It consists of thin layer of peptidoglycan, bilayer membrane outside the peptidoglycan contain phospholipids, protein and lipopolysaccharide. May be impediment to the treatment of disease, and it appears pink following the gram staining procedure. Archaeal cell wall. They do not contain peptidoglycan. It instead consists of variety of specialized polysaccharide and protein. In the gram-positive archaea stain purple, in the gram-negative archaea stain pink. Next, septum. It is a collection of a dozen proteins that localize to the division site, which they directly assemble in the division process. That is called septum. Segrosome. It is a multiplication complexes that partition chromosome or plasmids in the bacteria. A cell division process, it involves the cells divides into two cells and produces the daughter cells. There are two types of differentiation in the bacteria. One is single cell differentiation and another one is multicellular differentiation. Bacterial morphological plasticity refers to evolutionary changes in the shape and size of bacterial cell. As bacteria evolve, morphological changes occur to maintain the consistency of the cell. However, this consistency could be affected in some circumstances and changes in bacterial shape and size. In the bacteria, transformation into filamentous organisms have been demonstrated recently. These are survival strategies that affect the normal physiology of bacteria in response to factors such as innate immune response, predator sensing, quarantine sensing, and microbial signs. Bacteria have different shapes and size, which include coccus, rod, and helical or spiral shape. This form basis for their classification. Rod-shaped bacteria attach more readily in environment with shear stress. Example, in the flowing water. Coccus may have access to small pores, creating more attachment sites per cell and hiding themselves from external shear forces. Spiral bacteria combine some of the characteristics of cocci, small footprints, and of filament, more surface area on which shear forces can act, and the ability to form an unbroken set of cells to build biofilms. Several bacteria alter their morphology in response to the types and concentrations of external compounds. Bacterial morphology changes help to optimize interactions with cells and the surfaces to which they attach. This mechanism has been described in bacteria such as Escherichia coli and Helicobacter pylori. Oxidative stress, nutrient limitations, DNA damage, and antibiotic exposure are some stress conditions to which bacteria respond, altering their DNA replication and cell division. Filamentous bacteria have been considered to be overstressed, sick, and dying members of the population. Filamentous E. coli can be up to 70 μm in length and have been identified playing an important role in pathogenesis in human diseases. Nutritional stress can change bacterial morphology. The most frequent shape alteration may be filamentation triggered by a limitation in the availability of one or more nutrients. For example, Actinomyces israeli grows as filamentous rod or branched in the absence of phosphate, cysteine, and glutathione. However, it returns to a regular rod-like morphology when adding back these nutrients. Relatively few bacteria carry out a defined morphogenesis other than cell division during their normal life cycles. Still fewer bacterial species both differentiate and offer in genetic and biochemical manipulation. The criteria make use of bacteria reasonable the mode system for cell development, and it includes simple and well-defined differentiation in the synchronized cell population. Differentiating bacterial study include Bacillus, Streptomyces, Arthrobacter, Myxobacter, and Caulobacter. A rod-shaped bacteria Bacillus differentiate by heat-resistant and resistant spore turns vegetative rods through germination and growth. Sequential changes occurring in the sporulating cell have been carefully defined. It includes morphological alterations, alterations in the electron transport pathway, and the synthesis of spore-specific substances. Analysis of mutant organisms have demonstrated that genetic determinants govern that morphological and biological changes. During the outgrowth, the first gene product is new messenger RNA, followed by the formation of proteins and enzymes. This polymerase differs from that of vegetative cell in that beta subunit of the enzyme is altered with a generation in the template specificity. The Bacillus offer important guidelines only to the study of differentiation of bacterial cells, but perhaps of eukaryotes as well. Other groups of bacteria include Streptomyces, Myxobacter, Arthrobacter. Of these groups, Streptomyces can be grown on defined media and are particularly for genetic study. Hopwood and Semonti extensively studied genetics of these bacteria. The life cycle includes spore germination to form vegetative cell that develops into complex medium, then septate into aerial hyphae, and then produces single spore that initiate the differentiation cycle. On the other hand, studies with myxobacteria have emphasized the isolation of morphological mutant in order to define macromolecular behavior during morphogenesis. The bacterium can include induced to change from rod to microcyst form synchronously, but it cannot be grown on defined media and a genetic system have not yet been developed. Arthrobacter presents an advantage distinct from Streptomyces and Myxobacteria, the simple differentiation process which consists of biochemically, morphologically well-defined sphere rod transition is undergone in the environmental control. And the next one is Caulobacter. Bacteria of Caulobacter genus are aerobic gram-negative organisms like Pseudomonas metabolites hexoses via the Etner-Doudoroff pathway. A unique feature of Caulobacter bacteria carry out obligatory well-defined morphogenic changes during their life cycle. The Caulobacter are distinguishable morphogenetically in which they have stalk of one pole of the cell. The wall of the stalk is continuous with both the lipopolysaccharide and mucopeptide layer of the cell wall. Under normal growth condition, a new stalked cell synthesizes an intracytoplasmic structure at the pole opposite to the stalk, and this process represents the first step in the cycle of differentiation. The same type of membranous organelle is found in the stalked pole of the mature cells also. The second occurrence in the differentiation process is the formation of single flagellum and several pili also at the pole opposite to the existing stalk. Binary fission yield a sessile stalked cell and a motile swarmer cell. The daughter swarmer cell differentiate into stalked cell before it is capable of cell division. This transition make the bacterial at the point of attachment to the flagellum of the cell. The pro-existing polar membrane structure move the constitution to the interior of the new forming stalk, where the flagellum can be found as appendage of the tip of the stalk. Eventually, the flagellum falls off and can be recovered in the medium. Each step in the differentiation process therefore represents the synthesis of specific structure, that is polar membrane, flagellum, pili and stalk at the specific site in the cell and at the specific time in the cell's life cycle. Heterocyst in cyanobacteria that comes under single cell differentiation. In the presence of a source of combined nitrogen such as nitrate or ammonium, Anabaena grows as long filaments containing hundreds of photosynthetic vegetative cells. In the absence of combined nitrogen, it produces heterocyst every 10 to 20 vegetative cells along filaments. There is also a cyst formation that is initiated in the exponential growth when it undergoes a cell division, example Acetobacter. In the Myxobacteria, which is a gram-negative bacteria, spore-bearing fruit bodies are formed during starvation when the growth gets arrested. Swarming is a fastest known bacterial mode of surface translocation and enables the rapid colonization of the nutrient-rich environment on host tissue. In the Proteus mirabilis, swarming is an elongated, multinucleated and hyperflagellated spread, multicellular rafts across surface on high agar medium. In the Actinomyces isirelli, which is already seen, there is a filamentous rods are branched in the absence of phosphate, cysteine or glutathione. However, it returns to a regular rod-like morphology when adding back these nutrients. Next one is virus differentiation. Viruses are the proteins on the surface of nucleocapsid or envelope that become apparent and in which allows for morphologic differentiation. Viruses often have complicated life cycle in their natural host, replicating in the series of specific tissue in a defined order that may even vary depending upon the route of inoculation. These viruses show distinct preference for efficient replication in selected differentiated cell types and has been termed as tissue tropism. In order to understand the differentiation, virus factories or virosomes are created and nascent crescents are formed and engulf the unit genome. Immature particles are spherical but condense down to a dumbbell-shaped intracellular mature virus in a pox viridae. That is the first genus family of the virus. Second one is Viridae. The genome consists of 10 segments of linear double-stranded RNA. By negative strain electron microscopy, virions are approximately 85 nanometers in diameter, are roughly spherical and possess a double-layered protein capsid in a alpha retrovirus and gamma retrovirus. And third genus family is U-shaped at the circular particles. In the genus cytomegalovirus that belongs to a family, Filoviridae, it is linear double-stranded DNA. Replication takes place in the nucleus where nucleocapsids are formed or surrounded by an inner integument and travel to the cytoplasm by budding upon the inner nuclear membrane, passing through the perinuclear space and fusing with the outer nuclear membrane to egress to the cytoplasm. The next one is fungal differentiation. Under form specific favorable condition, there is the presence of asexual and sexual spores that to germinate into either unicellular yeast or filamentous mycelium. Most of the fungi differentiate generally in three phases: vegetative, asexual, and sexual. Fungal differentiation also includes the vegetative forms, that is hyphae and yeast, as well as specific morphologies to facilitate mating. Example, protoperithecin, plant tissue invasion, example, aprosoria, and airborne dissemination, example, conidia. The next one is protozoa differentiation. In most protozoa, the cytoplasm is differentiated into ectoplasm, the outer transparent layer, and the endoplasm, the inner layer containing organelles. The structure of cytoplasm is most easily seen in species with projecting pseudopodia such as amoebas. Two main kinds of morphogenetic events occur among protozoa. One is reproduction, process of reorganization including encysment, excisement, sexual processes, structural and physiological adaptation exhibited in the course of complex life cycles, and restorative events elicited by environmental stimuli. Examples of such events are discussed with reference both own intrinsic interest and to their value in the study of cellular differentiation in general. Stomatogenesis enceliates provide a remarkable instance of extensive and rapid differentiation. The phenomenon may be studied to advantage not only in reproduction but also in the macrostome-microstome transformation. Other simple transformation in protozoa providing unparalleled material for experiments on cytodifferentiation under controlled condition or excisement and encysment, the amoeboflagellate transformation, and leishmania leptomonad change. The more complex transformations occurring in the life cycles of trypanosomes and in the appearance of sex under the influence of an insect hormone provide additional material of basic significance to an understanding of the differentiation in the protozoa. Thank you.